So first of all, just thank you for the opportunity today to present on cell and gene therapy manufacturing. I'm going to begin with a high-level overview of comparing the traditional drug manufacturing process to the autologous CAR T cell therapies. So, a lot of people would be familiar with the traditional manufacturing process in terms of synthetic drug manufacture, tableting, for example. This process is a very fast process in terms of manufacturing. It takes days to complete. The testing occurs at defined steps in the process. The product tends to be very stable because it's synthetically made. It has long shelf life. It's typically stored at ambient conditions at room temperature. And it's quite simple in terms of supply chain in that you can hold inventory at different parts of the process to help with demand for the customer. Just looking at autologous manufacturing or CAR T therapy, it has longer processing times. The patient has specific therapy, so it's customized to the patient cells, so very much individualized. The testing is continuous, so it's not at defined steps in the process, it's right throughout the process. It's time critical to treatment because of the urgency of the disease. The drug tends to be frozen storage, which adds complexity to the manufacturing process and supply. And it's micromanaged, so right through from when the patient is identified through to when they're actually treated. So that it's fairly heavy in terms of manual interventions. Just moving on to the next slide. So this slide is giving a high-level overview of the manufacturing process for gene therapy in particular. So starting with one vial containing approximately 10 million cells for approximately 3 weeks, they are supplied with fresh nutrients so they can multiply and expand in number. From there, then the cells reach up to 1 billion in number and they're transferred to a bioreactor for continuous expansion. The plasmid DNA is added to instruct the cells to produce viral vectors and that contain the therapeutic replacement gene. A few days later, the cells are broken open and the full gene therapy vectors, empty vectors and other components are released. So this part, steps 1 to 3 are the upstream, or what we refer to as the upstream part of the process. And then steps 4 and 5 are on the downstream side. So step 4 is now getting into the stage where we're filtering the product to make sure that it's pure and safe for the patient. So any empty vectors that lack any therapeutic replacement gene is eliminated from the process. And then the final stage is the aseptic filling of the product into vials. In terms of Zolgensma, the gene therapy Zolgensma, each order per patient is based on their weight. So it's not as specific as CAR T in terms of their cells used in the manufacturing process, but it is weight-based. So there is an element of customization of the product. So the key points, sorry, if we just stay on that slide, please. The key points are, in terms of gene therapy, they're small batch sizes. It's quite complex and difficult to scale up. The manufacturing process is 24/7 monitoring, and this gives us a surety of the product safety, purity, potency, etc., which is tested throughout the process. It has short shelf life, so 12 months shelf life. Typically for traditional products, it's 24 to 36 months. And again, there's complexity with how the product is stored. So in terms of Zolgensma, it's minus 60 degrees Celsius. Again, adding complexity in terms of the distribution piece to the patient. Next slide, please. So this is my final slide, and it just gives you a snapshot of the timeline from manufacturing through to treatment of the patient for Zolgensma. As mentioned previously, there's a 12-month shelf life on Zolgensma. 5 months of that time frame is used for the manufacturing, testing, and QP release of the product, again ensuring the efficacy and safety of the drug for the patient. Once the drug is released, then the supply chain speeds up because of the urgency of the patient and the criticality of their disease. We can't hold inventory because of the short shelf life, and because we have to do weight-specific dosing. So we then manufacture the secondary packaging into regulatory packs, with their specific EU requirements in terms of blue box labeling, EU FMD regulations, etc. And so that packaging happens within 48 hours along with the QP release. And then it's shipped out to the site of care between 24 and 48 hours, depending on the location of the hospital within Europe. So just to conclude, and summarize, the manufacturing of cell and gene therapies are quite complex in comparison to traditional methods. They target orphan drugs, so it's small patient populations, and then the time criticality is a fairly big factor due to the disease and the urgency to treat the patients. Thank you.